denature under boiling conditions. And so you might wonder, well, do they have enzymes, or how do they survive at the boiling temperatures? Well, they do have enzymes. They're still made of protein, just like our enzymes are. But essentially, they're heat-proof enzymes. If you think of this line as representing the backbone of an enzyme, remember the concept that it has to hold its shape to function correctly. Your enzyme, or my enzyme, or just a regular bacteria's enzyme, is held in that shape by interactions of the R groups along the backbone. For instance, there might be a positively ionic charged R group here and a negatively ionic charged R group there. And so by the electrostatic attraction, by the ionic bond, it holds the enzyme in shape. But if you boil it, the thermal energy is enough to break this ionic bond and the enzyme denatures. Well, in archaea, the shape of the enzyme is reinforced by many more R group interactions, and so it essentially welds the enzyme into the proper shape. It's a heat-proof enzyme because of all those extra R group interactions. Matter of fact, this enzyme probably would not work very well at room temperature. It's more designed to function only at those really extreme temperatures. All right. There's another type of archaea they find called the acidophiles that live in extremely acidic pHs, like around pH 2, which is about the level of stomach acid. And you find some of these low pH areas naturally occurring in various regions of the world. Some places have natural sulfur deposits in the soil, and some of that sulfur is able to be transformed into sulfuric acid by natural processes. And so you do get these very acidic pools of water, much too acidic for most organisms to live in, but these acidophiles do fine there. As a matter of fact, in some areas you have a combination of these two, where there's a boiling hot spring and the water is like stomach acid pH, and you still find some archaea that are able to live under those boiling stomach acid conditions, so to speak. Here's another one. There are some archaea that are called the halophiles that live under extreme salt conditions. Now, first you might say, well, that's not a big deal. I mean, a lot of things live in salt water. Well, I'm talking way, way more salty than regular old salt water. Areas that are 5, 6, or even 7 times higher salt concentration than regular old ocean water. And usually you find these super high salt areas where there was ocean water, but a lot of it evaporated. When water evaporates, it leaves the salts behind. So as the water leaves, the remaining water gets saltier and saltier. The Dead Sea over in the Middle East is an example, or the Great Salt Lake in Utah are examples. These are salt evaporation ponds. This is where they let ocean water into shallow ponds and let the ocean water evaporate, and it leaves behind the salt, and then they collect that and grind it up and purify it, and they turn it into table salt. So this also creates a super high salt environment, and so you find some of these halophile archaea starting to grow there, which is incidentally why they have these weird colors. Some of these halophiles have some pigments in them that gives the water this funny color as it's evaporating. Now don't let that weird you out. Don't think, my God, if I eat the salt, am I getting a bunch of prokaryotes inside me? Well, they purify the salt quite a bit, and archaea don't cause any diseases. Some bacteria do, but archaea don't cause any human diseases. Oh, so here's a kind of a review question. Imagine this is just a regular old cell, not one of these halophiles. If I put that regular old cell in a super high salt environment, what's going to happen to it? What's going to kill it off? Water going in or out? Out, yeah, that's right. The principle of osmosis says water is always going to follow solutes, and so most organisms would die off in a high salt environment because all the water is going to get sucked out of them. In these halophiles, what the halophiles do is they internally generate a bunch of extra solutes, some sort of organic solute that they manufacture out of, well, organic molecules inside themselves. And so they fill themselves up with this super high concentration of organic solutes, and it ends up counterbalancing the high salt solute concentration outside themselves. So that they end up being isotonic to their super high salt environment, so no water loss. And one more example of the methanogens. These are archaea that can only exist in almost 100% anaerobic environments, meaning essentially no oxygen at all. As a matter of fact, they're so sensitive to oxygen, they get killed off if they're exposed to regular old oxygen environments. Now, where do you find environments like this? Well, even regular old ocean and lake water has too much oxygen dissolved in it to allow methanogens to grow. But in very, very stagnant waters, particularly the mud and muck at the bottom of like a swamp or something like that, the real black mud at the bottom becomes so stagnant and so depleted in oxygen, you get down to the essentially zero oxygen, and methanogens can grow there. Matter of fact, if you ever visit like a bog or a swamp or something, sometimes you see little bubbles coming up out of the water, and you may think, well, gee, is that a fish or something burping underwater? Well, a lot of times it's not. It's a bubble of a gas called methane that these methanogens make, and it bubbles up out of the muck at the bottom and comes to the surface. Matter of fact, I should mention that how do the methanogens get their energy? Well, obviously not through aerobic respiration because there's no oxygen there, and they don't even use anaerobic respiration. They have a special type of respiration which is unlike either of those, where they break down decomposing plant and animal wastes, and as a byproduct they make a flammable gas called methane. That's the stuff that bubbles up in the swamps. And in your intestines, you have some of these methanogens also. There's so little oxygen inside your intestines that methanogens can live in there. And not to get too rude or something, but when you pass gas, that's what's coming out of you. It's this methane gas from the methanogens living in your guts. And there's when we go to the bathroom, the feces contains a lot of these methanogens, and when it eventually ends up at a sewage treatment plant, methane gas continues to come out of it, and some of these sewage treatment plants burn the stuff off. You can't really see it in the daylight, but in some of these places, if you drive by at night, you can see what looks like a huge blowtorch burning away. Well, what they do is they suck off the gas from the feces and just burn it to get rid of it. I took a field trip to one a few years back, and I asked the guy, wasn't that kind of a waste of a valuable energy source? And he said that they do use it actually to heat up some water. The plant provides its own hot water from the gas that they're burning off.
So these don't use
They could break down glucose and other materials from dead plants and waste and things like that. So they can't use glucose. But it's I guess I'd say they break it down in a different way than we do, and they somehow end up with methane, just the same way we end up with carbon dioxide as a waste gas from the process. They end up with methane. And I'm not exactly sure of all the biochemical reactions involved, but I seem to recall that the sulfur atoms, which you find in some of the amino acids, get involved in the biochemistry.
But the basic general principle is that they still use glucose?
Well, yeah, but I'm saying that they're probably
differently, but they're still using
they do use glucose, but they probably also use a lot of the amino acids as well if they require sulfur. If I'm remembering right about the sulfur atoms, glucose doesn't have any, but some amino acids do. All right, and it's not only sewage treatment plants in garbage dumps, landfills. When the landfill gets filled up, they cover it up with a layer of dirt, and so they essentially create an anaerobic environment under the dirt. And a lot of these garbage dumps also do a similar thing where they suck methane gas from out of the decomposing garbage and burn it off. And it's kind of a neat thing I read about. There's this, I don't know, legend or ghost story or something you might call it, called the Will-o'-the-Wisp, and it's supposed to be some glowing monster or ghost that lives in swamps, and the thing is supposed to attract people who are curious about it into the swamp at night, and then they drown in the quicksand. And they think this legend essentially came from methane gas bubbling up out of the swamp and somehow lighting on fire. So it creates methane, it makes sort of a bluish flame when it burns. And so maybe static electricity or a lightning strike might get it burning, and it could conceivably burn for years out in the middle of a swamp somewhere. So if you're ever visiting a swamp at night and you see some sort of ghost-like thing, don't say, oh my God, it's the Will-o'-the-Wisp. It's probably just the methane burning off. Okay, those are the archaea. Let's look at the bacteria now, the other domain of the prokaryotes. And as I said a few minutes ago, bacteria, you can actually define bacteria and the archaea being distinct from each other by looking at the actual molecules that they're made out of. The cell walls and the membranes are very different from each other. But again, I just kind of took the easy way out here and defined them as what the environments where they live in. That the archaea live generally in extreme environments. The bacteria live in all sorts of environments, maybe not quite as extreme as the archaea do, but in quite a bit of diverse environments as well. But focusing on some of their biochemical distinctions, bacteria have a type of cell wall that's distinct from themselves. It's different from the cell walls that you find in archaea, and different from the cell walls that you find in plants and fungi and protistans. Now you remember that plant cell wall is cellulose. The bacterial cell wall is a substance called peptidoglycan, which you don't find in any other domain. Let's see. There it is. The purple part right here is supposed to be the peptidoglycan cell wall of the bacteria. And just like we saw with plants, the cell wall is outside of the plasma membrane. And I stole this from another textbook. This shows the structure of this peptidoglycan stuff, the cell wall. The G's are glucose sugars, and the M is a different monosaccharide. I want to say muriatic acid, but I don't think that's right. But anyway, it's a different monosaccharide. And so the basic structure is alternating glucoses and this M monosaccharide alternating with each other in long chains. So that part is sort of like cellulose, except it's not pure glucose. It's glucose alternating with this other sugar. But what really makes the peptidoglycan distinct is this. The chains of carbohydrates are cross-linked to each other by very short peptides on the order of around 10 amino acids long. And so it's a weird kind of molecule. You don't find in most organisms peptides and saccharides linked to each other in the same molecule, but you do in this peptidoglycan. Matter of fact, that's where the name comes from. Part peptide and part sugar, so peptidoglycan. And incidentally, some of the antibiotics, which are these medicines they use to kill off bacteria that have infected us, some of these antibiotics work by disrupting the peptidoglycan cell wall. Penicillin, for instance, breaks up these crosslinks, these peptide crosslinks between the chains of saccharides, and that weakens the cell wall so much that the bacteria can fall apart. The thickness of the cell wall varies quite a bit between different species of bacteria, And as a matter of fact, that's used as a classification system. There was an early bacterial researcher named Christian Graham, and he invented a stain, kind of a purple-colored stain, that could stain this peptidoglycan cell wall. And he found with some bacteria, they stained a very deep purple. Those were ones that had a very thick cell wall. And other bacteria stained only very lightly, ended up with sort of a pink color to them. And so those ones are called gram-positive if they take in a lot of the stain because of a thick cell wall. And the other type that only stain very lightly are called gram-negative, and they look pink under the microscope. If you ever take a microbiology class, this is probably one of the first ways of analyzing the bacteria that you study that you'll learn about. All right, what else about bacteria? All of them are unicellular, you know that. But that being said, some of them under the microscope you would see are filamentous, where the cells are linked together in long strands. This is an example of one you see right here. So here's an individual cell, and here's an individual cell, and here's an individual cell. But at first it looks like they're multicellular. They appear kind of linked together in these chains. Well, they do attach to each other. There's kind of a slimy layer that holds them together. But each individual bacterial cell could survive